Understanding how diseases begin and progress at the cellular level is one of the biggest challenges scientists face today. With the rapid rise of multiomics studies, researchers are combining advanced tools to answer one of life's most profound questions: what are actually we made of?
I was passionate about discovering how cells mechanisms come together and work.
But as we are discovering, biology holds complexities that can't be solved by only one field. In today's episode, I'm joined by a structural biologist with a passion for making science exciting and relatable. And he's here to help us to see why protein structures hold the key in understanding health and disease.
Understand
I get what you mean. So for many people who are doing strength training, muscle building, for them it's just protein one way to build the muscle.
Absolutely, yes.
Right.
So they get excited when a scientist talks about proteins because they think, okay, this is the quickest way to get muscular mass.
Today we will explore how these molecular blueprints within our cells could unlock medical breakthroughs of tomorrow. So if you're ready to dive into the hidden world of proteins and what they reveal about how our cells work, then just stay tuned. This conversation will take you behind the scenes of science, blend the beauty of biology with the power of innovation. So let's get into it, have fun and enjoy our conversation.
Daily dose of honest science.
Hello guys and welcome back to Daily Dose of Honest Science. I'm your host Dr. Chandni Kumar, and of course you are here for the best medical science podcast that you can hear on any podcast. Today I am joined and honored to introduce you to Dr. Leonardo Feletto. Leo earned his PhD in structural biology at the University of Leicester, exploring protein structures that shape gene expression and chromatin organization. Now he is a teacher in Italy and has an incredible talent for breaking down complex topics. Beyond the classroom, Leo's also passionate about science communication and even mixes in some break dances to make science even engageable. Please join me in welcoming Dr. Leonardo Feletto to the podcast. Hi, Leo.
Hi, Chatney. Hi, I'm so happy to be here and welcome to everyone to this new episode.
Thank you so much.
You told you told the audience so many things about me, and I'm a bit flattered. I hope I will meet their expectations, and we can discuss science in an interesting way. Finally, it's always a Monday afternoon, so I'll try my best. We're recording on Monday, so it's gonna be hard.
Yes.
So please stay tuned and support us.
Sure. So Leo, as I said, you are now a teacher in Italy, but of course, I would like to know what brought you into science. So that's like the typical question I want to ask.
On a Monday afternoon.
On a Monday afternoon.
Cool. I guess what brought me into science was the fact that I started asking questions about how things work. Of course, as many young students, I was passionate about discovering how cells mechanisms come together and work. And so this world was so interesting. I started asking myself questions about the inner mechanisms of cells, and then I ended up in a lab, trying to find out how did they work, and all the things that were hidden inside our cells. So that I think everything started like that. Also, I found out when I was like 20 years old, and I had to choose a university course, that the only thing that still fascinated me on a visceral level, I would say, was really studying biology. So that's why I started this route that brought me here more than more than 10 years later, I guess. So yeah.
Wow.
It's been a ride. It's
Amazing.
been a
It's
ride.
been a
But then
while.
finally, I think it's something that happens to any young scientist that really finds out this is what they want to do in life,
Mhm.
and so they start asking themselves questions. So I'm not. I wouldn't say I'm different from any young scientist that is really passionate about these
Mhm.
questions and want to know more.
Mm,
That's
mm.
it.
Do you have a particular topic that you are very passionate about? Because science is a
Well,
very
I went into structural biology just because. I studied structural biology at the very end of my university courses, and I was lucky enough. I found this course that was this structural biology course that was not really popular at that
Mhm,
moment. So I
mhm,
was just standing there together with a couple of other students, and we were interested, but at the same time we knew the topic was going to be difficult. And at the same time, I think I was there right at the right time, because it was 2017, and that was the time in which cryo-EM was kind of the trending topic.
yeah.
So as soon as I realized I could join a lab and using these new techniques, well, at least cryo-EM used to be a thing since the 70s. This is
Mhm,
a technique that uses this electron microscope to investigate the shape of proteins and other biological molecules. So as soon as I found out I could be involved in a research project that was, making use of this technique, I was
mhm.
really interested, and I just went along and started using these techniques, and at the same time using them to explore the inner life of cell.
Mm. Really, really cool. And I'm also fascinated by structural biology. Just for the audience, maybe the term is
Yeah,
not
no,
very
we,
credible.
we, we want to step back maybe and
Yeah.
explain a bit more about this technique and
Yeah.
how it
So first
works.
of all, structural biology is how our proteins actually folded, because they have to have a certain, yeah, let's say, um, shape. They need to be in a certain shape, let's say, so they can actually be functional. So that's really important. And we already know that if this is not the case, we have something like Alzheimer's disease or Parkinson's disease, because these proteins are not well folded anymore. About the disease mechanisms, there are many, but in theory now, also what Leo has studied is more like how does the protein actually do its function, and structural biology is a really, really important field to understand the function of a protein. And there are many, many techniques out there to address this. And as I said, cryo-EM, so cryo-electromagnetic, I guess it's called.
No, it's a
Cryo
cryo-electron microscopy. It uses
microscopy, yes.
cold temperatures
Yeah.
that are. I mean, you can get these temperatures in particular ways. Then we can discuss it further. But
Mhm.
the thing is that when you mention proteins, the way I usually explain it to students is that the cell needs some tools to
Mhm.
perform its activities, and it comes with a toolbox, and you use tools just to go through particular things. I mean, the way you use a hammer, the shape of a hammer,
Yeah.
it's literally made to stick a nail to the wall,
Yes.
and you wouldn't use anything else.
Mhm.
And the way you use a screwdriver, the shape is really connected to its function. So at the
Yeah.
same time, each and any protein you find in a cell, it's structured in a way it can perform a particular function. So, as you said, when you have diseases, it means a particular protein of your cell, it can be, impaired, and so it doesn't perform the function as in the right way and can cause it can cause problems. So, for example, Parkinson's diseases and all these things that can happen in cells sometimes. So you want to know the structure so you can eventually correct it and reverse the condition to a normal condition in patients.
True. Or understand why did it actually started to malfunction, right?
Yes,
So,
yes, of course.
So that's kind of the fascinating part. And to be honest, to look at these structures, it's like fascinating. It's so beautiful, right? And you have to imagine when you have such a structure in front of you, that is like inside a cell that you cannot see. And this is like crazy. It's like, wow.
Definitely. Also because we of course we see structures and we think of something that is still in that very shape and is always like that. But then in cells, all these shapes are coming together and changing all the time to adapt themselves to particular situations. So these molecular machines are built in a way they can adapt to different conditions. This is also, I mean, if we're talking about frontiers of structural biology, this is also something structural biologists are trying to understand. So how can these structures adapt to different cellular conditions? And obviously this is fascinating because it's like a hammer you have that doesn't only stick nails to the wall, but can adapt itself to many different conditions. I mean, and everything is ultimately written on your DNA. And of course, these proteins evolved so they can perform many different tasks according to many different situations. The whole concept is just mind-blowing.
Mind-blowing, yeah. I completely agree. And this kind of excitement that gets me all the time, like today was also like a day. And I'm so happy to have you on the podcast to share also the excitement.
I'm so happy to be here.
So, of course, I would like to go a bit deeper into what you worked on, because that would be also kind of interesting. So, can you share a bit more? What were your topics and what did you solve maybe in the end?
Oh, well, so I was studying the structural biology of some proteins that are involved in the structure that is within our nuclei. So in the nucleus of our cells, DNA, which is the code that we use to write, all the instructions that make our cells what they are, the DNA is tightly packed. So if we were to unwrap all the DNA that is inside each and every one of our cells, I mean, the DNA would be so long because it's tightly packed in the nucleus of our cells. The DNA would be so long, it would cover a distance of 2 meters, which is incredible. Because if you think to a tiny cell that is just a
few microns.
A few micrometers long, and you think that the nucleus is even smaller, and within the nucleus you've got all this DNA packed, 2 meters worth of DNA packed in the nucleus. So this is already incredible. And so scientists started asking themselves, how can this DNA, this amount of DNA, be packed in a tiny space, which is the nucleus? And they realized that there are so many proteins inside the nucleus that help the DNA to be packed in such a small space. So this structure made of proteins and DNA is called chromatin. And chromatin is exactly it's the structure that makes up chromosomes in the end. So the way you pack DNA is through chromosomes, and chromosomes are built through chromatin. And so during my PhD, I was studying some of the proteins that are helping chromatin to be built this way and help chromatin works. So the way they wrap around the DNA, it makes chromatin what it is.
Yeah. Awesome. Can you go a bit more deeper, like on what
Yes.
kind of proteins you worked on, or?
Sorry about. I hope you will be able to cut. I just received a call, and
Okay.
it shouldn't be a problem. Hopefully, I should have switched off my phone. I'm sorry about
Mhm.
that.
No, no,
Um,
fine.
I'll switch off my phone. Sorry. Yes. So, what I studied during my PhD in particular was a bunch of transcription factors that sum up. I would need to explain what are
We are
transcription
going
factors
this
first.
time very
Okay,
deep. Okay. Yeah.
a bunch of proteins that we can see as so. If we imagine DNA as a book filled with instructions that are needed for the cell to, be what it is. So the DNA is a book, and there are some proteins that are used as bookmarks. These bookmarks can come together and highlight some parts of our DNA that are important to build some parts of the cell. And during my PhD, I studied a group of these bookmarks that are called transcription factors in our cells. So the idea of my PhD was to sum up all these bookmarks to form a unique structure and to look at that under the microscope. Then, of course, it was kind of an ambitious project. So at the end, what I did was just to look at some parts of these proteins, and it was already challenging to understand how these parts were interacting with DNA and understand how do they work. But finally, the idea and the what we learned from my PhD project and what it would be interesting to find later would be to understand how this whole thing interacts with the with the environment in the nucleus and how this regulates the reading of the DNA, how this regulates the way DNA is read, and how these instructions finally come to build many different proteins outside the nucleus.
And I think it was all like in vitro work, or did you also had some in vivo work?
No, it was all in vitro. It was all in
Wow.
vitro.
Okay.
Because let's say this is like fundamental research. So finally, you can apply all these things to, some treatment, some drug designing project. But at the very beginning, you want to understand the biology that is at the foundation of all these mechanisms. So what I was doing was really studying the foundations and the fundamental sides of biology in order to understand how these things work and then use them to design drugs or something like that.
Did you follow up your work? Like,
Well, at the
did
moment, I'm still
someone?
in contact with my former research group, so I know what they're
Good.
studying. And
Nice.
no, I was actually in contact with some PhD students that are still studying what I was studying,
Oh,
and
wow.
was a bit excited to learn a couple of weeks ago that they're ordering new constructs to start from where I left my
Cool.
project, and that is cool because it's like a story that goes on. Then, unfortunately, I left research, so I'm not into it anymore. But I'm still happy to know the story is going on and
Mm, mm,
it's brought forward
mhm.
by someone else.
That's wow. That's so cool to know, and also that you stay in touch still, right?
No.
Uh,
Well, what can I say is that during the time I was doing my PhD, it was it happened during the pandemic years.
Yeah.
So I spent I spent my PhD in the lab in a time I couldn't really move from the lab because outside everything was just shut down. I mean, we were doing that during the lockdown. So this helped me build some strong bondings with the scientists that were working with me in those years, and they're still good friends. I'm still good friend. They're still good friends I can rely on,
Wow.
and I'm still in contact with them.
Nice. That's really, really cool. As you said, you're not in academia anymore.
No.
You're a teacher now. So,
Yes.
can you just describe a bit like that's a huge transition, first of all, becoming teacher. And then what kind of subjects do you do now, right? So,
Well, of course, I'm teaching science, but at a level that is a bit, I'm teaching teenagers. So, first of all, I moved back to Italy. I was working in the UK, and I moved back to Italy to teach teenagers, which is kind of like, stepping back from something completely different because once you start doing research, the expectations are like, okay, you'll just move on and do something even greater after that. I don't know. No, I just came back and started my teaching career, and so what I did after my PhD, I was really fascinated by science communication, and so I wanted to find a job in the field, and I started looking for a job in the UK. But then Brexit happened, and a lot of things came together at the end of the in 2022 at the end of the pandemic, and so since I had already started a teaching induction in Italy before moving to the UK for my PhD. So I just came back and went on with that, and I happened to get a position as a teacher. So at first I thought, okay, I'll teach for some time until I won't be able to find a job as a science communicator. But then I just stuck to it, and what I'm doing at the moment is teaching, and as a side hustle, I'm going on with science communication projects, and I'm still involved in some science communication projects in the UK, and I'm also following some science communication projects in Italy. But yeah, most of the time I'm just teaching science, and well, I realized teaching is somehow a way to communicate science, even though there are some differences. And if you want, we can go deeper
yes, please. I would
about
like to.
that.
Yeah.
No, because of course the way the public looks for science and wants to know more about science, so the public searches actively for science communicators is different than what you've got in schools, because of course students cannot really choose, and they've got a teacher, and the role you have as a teacher is a bit different, because of course you need to adapt concepts in a way they are understandable, but at the same time you need to evaluate their understanding. So in the end, it's different from communicating science in a podcast or in another space on social media, for example, because you give your audience a concept, but somehow you're not evaluating them. You don't have to be a teacher, right? So you're not there just making sure they understand. You're just there to transfer excitement
Yeah.
and communicate, the new stuff that is being discovered in science. But that's it. You just need to perform and to to do it right, so they understand. Teaching is a bit further, because you need to make sure that what you've communicated is understood, and they understand, and I don't know, an audience can choose a science communicator, while students cannot choose their teacher.
Yeah.
So as much as I like to communicate science in class and online, for example, I can really understand how these two missions are separate and they're different. But both a science communicator and a teacher do that for the sake, for the sake of the fact because they want people to be educated about science, and they feel the urge to transfer information and excitement about what we are passionate about, basically. But yes, these are two different missions, and let's say I tried to find a compromise. Since I didn't find a stable job as a science communicator, and yes, I was involved in short-term projects, but I was never able to find something for the longer term, while I was able as a teacher, and I am using my skills as a science communicator to do this for a living. Then who knows in the future? I mean, I can see the positive sides as a teacher, and there are other positive sides as a science communicator. Maybe in the future I will move more in one direction or another, but that's I'm sure that this is what I want at the moment, and let's see in the future.
That's so nice to know that you kind of found your way, but I would like to know um, how. So if you if you do your teaching, of course, it should be exciting, but also a bit humorous, right? So no one likes a teacher that just stands there in a monotonous voice and telling.
No, absolutely, absolutely.
So
Especially, new
so what are your
generations, they
concepts?
want to be entertained.
Yes.
They really want someone that stands up and, you know, tries their best to make things vibrant and active. And at the same time, it won't be enough, because students know that this person is there just to, to evaluate them. So they will always be a bit. You need to bring out passion from them, also because you want to motivate them to, go through a particular career later. I mean, my students are young and they still need to go through high school. But then ultimately they need to find what motivates them in life. And so it's kind of hard to see them every day and come there always with trying to transmit this feeling. I think this is really the challenge for a teacher. While a science communicator, science communicators speak to a broad audience, and of course, the audience they speak to, there will always be a younger part of the audience that want to learn, just to, to imagine their future in science, or something like that. But most of them are also, most of the audience is made by adults that are just curious and want to know more just because they're interested. But they already made different choices in life, so it's a bit different. You need to adapt your speech or whatever, or your presentation, the way you build up concepts and the way you want to transfer them, so so you make sure they reach the right that they reach the public in the right way.
Yeah, I see it the same way. So for me, it's as a postdoc right now, I'm um, involved in teaching, but my audience is the medical people. So like medical students in their very first years. So they don't have clinics, they have the preclinical phase. So they learn all the concepts like what is transcription, what's translation, and all these kind of
Yes.
things.
So I'm
But
sure with an audience like that, you can already, there
yeah,
a bit more, and you can
yeah.
use a language that would be impossible for me for the age of my students.
Yeah, yeah. And but I still try to adapt myself because it would be boring to just come with the concepts if they don't understand why this is maybe important for later. So I try to dig a bit deeper, finding maybe diseases that are associated with each mechanism, or just that they have something to grab on that they can understand. So I completely get what you are saying, that for your audience it might be a bit too much. They will be overloaded, they will not
Yes.
understand. And for you, as having one heart on the teaching and one side of the heart is for science communication, then of course it's a bit difficult for you. Which side is now on the upper level to
No,
actually?
it's always a balance. It's like
Yeah.
in chemistry, you need to
Yeah.
balance out the
Yeah.
reaction, I guess.
Yes.
You need to balance the reaction out every time, every day. Yeah, no, it's at the moment it's exactly this. At the same time, another thing that makes teaching a challenge sometimes is the fact that as a teacher you have high expectations coming from parents. For example, and the trust parents um, give and give to science, and the fact they expect their children to be good at science or scientific disciplines. We live in an uncertain world at the moment. So if there's one thing that could grant children a better future, then parents would expect their children to be good in these subjects. So maths, science, and all these things. And so they come to me and they kind of ask me, what can they do to make their children passionate about what I teach? And sometimes my answer is that let your children just be passionate about what they love. That would be the best choice for them, because in the end, even a career in science can be hard, and you go through that only if you're really passionate about what you're doing.
Yeah.
From the outside, there's this thing that many people think that in science people just find jobs,
Yeah,
but,
but
but,
it's
but
not
of
like
course
that.
it's different, right? So
Totally.
you don't really. I don't think there's an easy path in our world. You just need to be passionate about what you're doing,
Yeah.
and that's it. And then everything will come later, somehow, in some shape, in some form,
Yes.
like proteins. And then of course they can change and they can
Yes.
adapt, and you need to have these skills in today's world. And
Yeah.
so this is also what I try to transfer to students. It's not important if you're not passionate about maths or about other things, as long as you try your best. You need to find what really passionates you and go through that way.
Yeah. No, that's completely true. And I can see from your excitement that you're really passionate about this, telling people how it actually works. What I'm a bit surprised at, as you said, because of all the circumstances and the Brexit, you actually got back from England to Italy. So you were out from Italy, I guess, right? So
Of course, no. During my PhD, I was living in the UK. I did my PhD in Leicester, which is this city in the middle of England,
Mm-hmm.
and I made friends there. I'm still in contact with them,
Mm-hmm.
and it's always fun to come back and visit them.
Mm-hmm.
But then, of course, I tried to find a job in the UK,
Mm-hmm.
and I was involved in many projects in the past few years. You know,
Mm-hmm.
short-term projects as a science
Mm-hmm.
communicator. But then, no, in the end, I came back to Italy. For many reasons, because of
Mm-hmm.
Brexit, because my family was here,
Mm-hmm.
and let's say, for the moment being, I decided it's okay like that.
Mm-hmm.
But then, of course, I'm always excited to be involved in projects. Let's say, ideally, in the future, I would be happy to find opportunities that can, take me out of Italy
Mm-hmm.
for some time,
Okay.
even remotely, to be involved in project. Because of course, when you do
You
a
are right now
uh,
in Germany, so
yeah, when you're
that's
involved
kind of
in a research project in an
yeah,
international environment,
yeah,
then you can never come back. Come on, it's it's so fascinating, because you
hmm
meet so many people from many different countries, and
yeah.
this is probably one of the things I miss the most.
Oh,
While I'm here living in Italy. I mean, Italians are okay. But first of all, I'm a bit strange as an Italian. I
really?
guess
Okay.
no, I guess many people from Italy would look at me and they're like, oh my God, from where,
Okay, I will
where,
ask my colleague then. He is from Italy, Marco, so I will ask
no,
him
but
if
but of
you're real.
course, this colleague of yours is living abroad, so he's already changed. But many Italians that live in Italy and
Yeah.
just stay there their whole life, they don't really understand that people outside change. And
Ah,
so when
okay.
I came back,
Okay.
they're all like, oh my God, this guy is completely crazy. What is he doing? What is he saying? But then, I mean, of course, I think I'm fun in the end. I don't know.
For me, it was a bit of a surprise because you have such an English accent. So that's why I was like, okay, how does it fit that you're from Italy? So
No, no,
that
but my English friends tell me that my accent is really Italian, so I guess I believe them. I don't know.
okay. For me, it sounded a bit more British, but okay. Um, just on this note, so do you see a difference from England and Italy in a way of communicating science? Do
Yes,
you?
yes, absolutely. I was really fascinated by the way, there's an English tradition for science communication.
Oh.
They really started this trend in the 50s.
Oh,
I would
okay.
say it's not only England, I would say it's the Anglo-Saxon world.
Hmm.
So in the US and many science communicators in England, like Attenborough, they
Hmm.
started a way to speak to people in a simple way that was then let's say copied, or many science communicators in Italy
Hmm.
were inspired by this work that was done by English science communicators. And these techniques, these hooks that are used to enthuse people and the audiences are still used, after this kind of tradition that was
Hmm.
led by English science communicators. So I could really see that. I could really see a different scene while I was living in the UK, and I came in contact with many science communicators there. Then, of course, I came back to Italy, and I'm in contact with the local scene. Um, uh, but yeah, I can see that even in Italy at the moment, there's a large scene of science communicators. Maybe what is missing is that in Italy it's a bit harder to get science communicators, science communication translated in an effective job. Many science communicators I know from here, they have just so many short-term projects that come all together. They manage as freelance professionals. They manage to build up a a network of contacts, and so they're in contact with so many people, and they manage to get many jobs all together and work in this way. While on my side, let's say I'm still trying to build up my network, and as I said, I was involved in short-term projects, but it's just not enough to, get a salary. So
Yeah.
that's also the reason why I thought it was going to be better if on the side I was going to have a stable job like teaching.
Yeah, no, that's I just had previously another science communicator from Austria. So many greetings to Martin. And he also
Ciao,
said
Martin.
like yeah, he is one of the bigger guys, I would say, and still, he's his own boss, but still, it's also not easy, and right now he can do what he wants, but it was also a bit, a lot of
No, it's hard to
yeah.
build up your own network,
Mm,
and to be sure that any time of the year you can fill up that gap with some sort of a with a job, so you can get the salary at the end of the day. Yes, or the alternative, what I was trying to do at the end of my PhD was trying to work for a research body as a science communicator in an institution. But then that really, well, I don't know, because I tried to look for a job in this way for I have tried for a couple of months, and then things weren't really working out. My salary ended, and so I just came back to Italy. And still, I was looking for jobs from Italy, but it was not the same thing. So I started applying for short-term projects, and I was luckier on this side. But then, yes, that really didn't work out at that time. So I just at some point I just got tired and started doing something else in between. But yes, if you so there are these kind of jobs. If you work for an institution and you do science communication for this institution, then of course things are better. Let's say they are better because you can work for someone else, but then it's not really you, right? Because
so
you're
you're
not
working
making
for
your
the
own content,
institution.
you're making content for someone else. So
Yeah. And you have to always find, this political language must be correct.
yes, yes,
You're not
absolutely,
allowed to
absolutely, yeah.
use. So I completely understand. So the kind of freedom you would like to have is not really there. So you're representing more the institution. So you need to
Yes,
take care of
absolutely,
that.
absolutely. So you need to pay attention to many different things
Yeah.
because I don't know, the examples you use, you need to discuss them first with supervisors
Yeah.
or other people who are there to tell
Yeah.
you what to do and what to say. So, yes, I guess one needs. Anything you do in life, you need to find a balance and
Yeah.
you need to find a compromise. So,
Yes. Yeah. So right now you have your compromise, and we hope.
yeah, let's say my compromise is okay. Has been okay so far.
Mhm.
Let's see how how long it works, and
Yeah.
then who knows?
Yeah, I'm looking forward to for new endeavors from your side.
Yes.
Um, but one thing, of course, is so you're not in academia anymore. But I, I don't know. From the conversation that we had, I have the feeling you miss the lab.
Yes. No. Well, I actually I actually dreamed once about the lab, and
Oh.
in this dream, I was trying to challenge. Somehow, I realized I was in a dream, and I tried to challenge myself to remember if in the dream I remembered the protocol to purify proteins.
Oh, wow.
No, I
Okay.
spent so many hours purifying proteins.
Yes. Yeah.
You can't imagine,
Pain.
and
Sometimes
so
a pain.
yes, no, definitely. And then it depends on the protein because there are some proteins which are a piece of cake. So you can
Yes.
just
Yeah.
purify tons and
Yeah.
it's easy.
Yeah.
But there are others. Oh my goodness. No, it's
Yes. Where you
completely.
have to try around to different
No,
constructs
different conditions and you
and
spend months on
yeah.
them. No, it's
And
crazy.
sometimes
No,
you
but
have to try different organisms. So sometimes you
yes,
do
yes,
bacteria,
definitely.
insect
And move
cells.
back
Yeah.
and forth from
Yes.
different. No, no, it's completely hell. As you know,
Yeah.
these are experiences that really leave you some. You know, they impact you somehow. So
Yeah.
that's why I'm still
Yeah.
dreaming about them. But
Yeah.
at the same time, I can tell you that I enjoyed the process because I was surrounded by nice people, and so this really made a difference, I guess. And well, as I said, I'm still in contact with my former lab, and I'm still happy to hear from them. And
Mm.
so, since I enjoyed my time in the lab, at the same time, I decided that I didn't want to pursue a research career
Oh,
because, of
okay.
course, I really wanted to. I enjoy maybe the side I enjoyed the most was the one bound to the the communication of my science and
Mm-hmm.
my results.
Mm-hmm.
I realized this was really what I enjoyed the most. And sometimes the lab can be painful. Of course, I missed that, but I really had to decide what I wanted to privilege at the end of
Mm. Mm.
my career as a PhD student. So I decided um, to leave the lab, and that's why I didn't go on with a postdoc. At the
Mm.
same time, I must be honest. When I finished my PhD, my publication was still not really there,
Yeah.
and so it was taking some time, and I wasn't really interested, and I simply went to. I went on with something different. At the same
Mm.
time, during my PhD, I was involved in some science communication projects
Mm-hmm.
in between, and I thought I could use that in a portfolio to to try to apply for science communication jobs.
Mm-hmm.
Then this thing really didn't really work out, but yeah,
Mm. Yeah.
I
Um,
tried.
it's difficult because I think right now people are acknowledging, so they start now to acknowledge that if someone is doing science communication, but I think some years before
Absolutely, yeah.
it was not really something. It's more like, okay, he or she has this weird side hustle, maybe, let's
No,
say it
no,
like
no,
that.
yeah, yeah,
And
yeah. No, I completely
now
agree.
people, especially after the pandemic, I think realize how important it is, and many things went wrong. And also Martin said that. Then in Austria, many things went wrong, also in Germany, and let's say the distrust in science also is something that is more and more coming, unfortunately, through social media, because so many people do not make the effort to really explain things properly, which is something I also realized back then. My career is a bit smaller, so like 1 year ago or something, but I really saw that we need better communicators,
No,
especially the
clarity,
ones.
honesty,
Yeah.
the same honesty that stands
Yes.
right
Yeah.
on the name of this
Yeah.
of this
Podcast.
podcast. So
Yeah.
yeah. Well, what can I say? My supervisor, for example, at the right at the beginning of my PhD, was a
Hmm.
bit puzzled by the fact I was spending my time out of the lab, getting involved in science communication projects. But I must
Hmm.
admit, at the very end of my PhD, once I established I wasn't really interested in going on with a postdoc. It was really supportive and helped me finding a couple of grants I applied to at the end of my PhD, science communication grants. So I'm really grateful for what
Wow.
he did.
Okay.
But I completely agree with you. Let's say maybe in the past it was rarer to find supervisors that were supportive
Yeah.
on this side. But it's true that succeeding in academia is increasingly difficult.
Yeah.
So I think even supervisors now came to realize that you need to find something else,
And
which
it's
can
part of
be,
the grant applications, right? So it's
yeah,
also
yeah, yeah,
something
yeah.
that they want to
Yeah,
be said.
absolutely. No.
Um,
They really put money into it.
yeah.
What I think it's important though is that not all researchers can be really, motivated enough and to do proper
Also,
science
well,
communication.
do it well, right? Not
Yeah,
everyone can
absolutely. Because
communicate well.
no, absolutely, because it's not a skill you need to possess as a researcher. That
Yeah.
it obviously help you, but it doesn't mean it needs to be there.
Yeah.
And that's the reason why I think many researchers should be, you know, should get in contact with a network of science communicators to
Yeah.
understand what's the best way to communicate
Yes.
effectively
Yes.
concepts.
Because also not watering down your science, right? Because
Yeah,
sometimes science communicators are not in that field, but they can help you, that you can,
obviously.
display
No,
your,
you.
right? So how do you see that? So
No, absolutely, I agree. And let's say we are getting there, but since it's been recognized as a separate mission only recently, um, let's say that the science world, scientific world is not ready to consider all these professionals and to counter them, to you know tell about what they're studying and to find the best way to communicate concepts
yeah,
and in a way not to be misunderstood when they're talking about their science.
yeah,
But we are still in the process, and at the same time, many times, some researchers who are not really ready or able to communicate science in the best way, they, especially if they communicate sensible topics, they I mean, we saw that during the pandemic,
yes,
they
yeah,
risk to be misunderstood and and then
yeah,
the effects can be unpredictable. So
yeah.
it's a fragile topic. You need to be careful about this thing.
Especially also about topics regarding longevity or
Yes,
aging,
definitely. Yes.
some particular stuff about
And
sleep
diseases. If you're
diseases,
talking about
Alzheimer.
cancer,
Yeah.
I mean, one thing I'm always telling this to students,
Mm,
for
mm,
example, because even them can understand the
yeah.
thing, is that when they use this when they talk about, oh, we finally cured cancer. No, you aren't. No, you're not. You're not curing cancer, and that's it. There are millions of cancers. You're finding a cure for curing one of them,
One of them.
or maybe a family of them. But there are so many different kind of cancers. You cannot possibly find a cure that is fitting them all.
Universal,
Le-
yeah.
yeah, let's say we are progressing towards an understanding of cancer as a process,
Yeah.
but a cure. Yeah, I know it sounds cool, and it's if you want to write an article or you want to get funds, it's useful, right?
Yeah.
Because you say, oh, we'll solve a decades-old problem
Yeah.
just with one snap. But no, it
Yes.
doesn't work like that. The
Yeah.
things are complex.
Yeah.
So ideally, you want to communicate complexity.
Yes.
But then, of course, it's hard, right? Even if we are talking about different topics like
Mm-hmm.
climate change,
Yeah.
it's so huge. There are so many
Yeah.
different things you want to consider.
Yeah.
One size definitely doesn't fit all. You
No.
need to help people, um, you know,
Navigate through this.
yes, absolutely. You need to help people understand the complexity
Yeah.
and unpack this
Mm-hmm.
complexity, get small bits,
Mm.
and understand how we are progressing by solving each and every one of them
Mm.
slowly.
Yeah.
But yeah, it's hard.
Just to give another example, so I made a post about the cure for type 1 and type 2 diabetes, which was a
Oh,
stem
okay.
cell
Yep.
therapy. And, people already, and unfortunately also from the science communication space, I've seen, they just took the Nature article, just, the screenshot
Copy and
of the
paste.
title,
Yes.
yeah, put it on their post and be like, we found the next cure.
No.
And also some not related people were making little posts about it, that this is kind of a new thing. And then for me, I wanted to give the audience or the people who read my post a bit of background, because many of the people don't understand what is the difference between type 1 and type 2 diabetes, right? So first of all, you need to make that clear. Then secondly, what is the main mechanism behind each of them? So, and why stem cell therapy? And that already, as you said, is showing the complexity of this whole topic. And it took me, like, I think I made 10 10 post things that you can carousel. And no one really took the initiative to dive deep into it and tell the people about that. And that is the
See,
problem that I see.
there's another compromise you need to find as a content creator. If you're active on social media, you need to tell people the truth, and you need to be you need to be complete. You need to tell
Mm,
them the whole story, but at the same
yes.
time, you need to catch their attention. And
Yeah.
when it comes to catching people's attention, then it's completely madness, because
Yeah.
the attention span on social media has been decreasing for years, and
Mm,
now it's we know that because we are active on social media.
yeah.
It's always so hard to communicate complexity and
Mm,
to get people attention at the same time. So
yeah.
many science communicators or many people who are active
Mhm.
on social media
Mm,
just try their best, and so since they don't succeed, they start doing smaller and smaller bits of science
mm,
till when the content is not even there, it's
yeah.
not clear at all, and they just come out with these titles that are completely
Yeah.
madness.
Yeah.
And yeah, you could say, there, you ruined it. But then the problem is that, I mean, I'm not really judging these people, because as I'm sure you understand,
Yeah.
some I want to see their good initiative, let's say,
Mm,
and I want to say that standing out on social media is sometimes is really difficult, and you try to use everything you can, but then you would need to do that in a way you're always honest with yourself,
mhm.
and you're doing the right thing, and you're giving people an idea of the complexity that is behind things.
Yeah.
I was talking about that yesterday night, by the way.
Oh,
I
okay.
posted a couple of stories, and my worry is that sometimes I see science teachers on social media
Mhm.
or other science communicators, well,
Mhm.
let's say science communicators,
Mhm.
who tend to mix a lot of things just
Yeah.
for the sake of you know,
I saw the story
yeah,
that yeah,
creating
yeah,
interesting content, but
yeah,
then it's not even content. It's just a couple of sentences you use to entertain people, giving them the idea or the impression you got it all. You're now a scientist. I mean, I know it's not exciting, but
yeah,
it's just a bit of the whole story, and
yeah,
what's behind that is really deep, is really complex, and you need to understand that one bit at a time. And so you know you need to study years to understand
yes,
everything, and
yeah,
at the same time you need to remember that there are people out there, science communicators or teachers or science journalists,
yeah,
who digest everything for you so it was ready and clear. So there's already a filter in
yeah,
there. You need to go back,
yeah,
read the original
yes,
article if you're interested, understand it. It's a long process, right? And the
and
idea
usually
would
no
be that
one is doing that, right? So
no, no,
if you would,
absolutely. But I wouldn't even expect my students, for example, to do that because it's not the. They're not supposed to do this. There are professionals who are supposed to do this, but they should give people who are reading their content the idea that everything is just more,
yeah,
right?
yeah,
And yes, no, this is in the best scenario in which people are honest, and then there are people who are not honest at all, and they're
yeah,
just writing, you know. Can I swear? No,
yeah, sure,
they're just writing bullshit
yeah,
just to sell people stories who are not even there.
yeah,
And then, well, I'm sure you saw a lot of these posts, and they're just there, or maybe they're not even made. They're not even made by real people,
yeah,
and they're just produced automatically by artificial intelligence
yes,
just for the
yeah,
sake of entertaining
yeah.
people. But that's not even science. That's just made up.
Yes, yeah, and that's this critical area where we need to be there to make the record straight so people understand what's going on. And I think it's really important to tell the people to be a bit more critical about every post and every reel that you see, especially now when AI is coming and can also produce videos, right? So that is, I think, a very, very important take home message here that people should not believe everything. And even if, let's say, a medical professional or anyone else is coming up with scientific stuff, it's always a gray zone area because in the end that medical professional might be not in working in research, right? And then how
Yeah.
this person should know how research works
No, I
specifically
think
for
just
medical students. Like they, they come and be like, "Hey, I've learned a lot," but in the end, it's not.
not at all, not at all.
Yeah.
I think first things first. Coming back to, talking about
Mhm.
school, people would need to think critically
Yeah.
and to evaluate what they've got, and ask questions to themselves and to the people that made the content about what's going on. Is this the whole story?
Yeah.
From where did we come to this content? What happened before? So it's a whole series of questions that really makes you understand if something is genuine or not. And in principle, these things should be taught at school. I try to help students understand these things at school. But then, of course, when school is over and you go on in life and you make your own choices, you need to remember, and you need to select
Yeah.
good content on the
Hmm.
internet. So at least, well, the algorithm finally is modeled by you and your choice and your own choices.
Yeah.
So
Yeah.
if you help the algorithm understand what's genuine content and
Yeah.
what's not,
Mm.
then it's for the best. It's for the best,
Mm.
and anyone will ultimately benefit
Yes.
even us.
Yes. Yeah. True. I want to before we go to our second part. Um, I just recently saw that you are a breakdancer as well. So let
No,
us
no, no,
know.
yeah, yes, no. This this traces back to even before I was well. I started breakdancing when I was 16 years old, so it was 2009, and no, it's a great passion I have, and I simply went on with it till now, and I use that since I guess it's a spectacular dance, and it's a part of my identity.
Yeah.
So I use that also to with science communication to explain difficult concepts in the
Yes.
past, and I'm still using it to this day.
Yeah.
And yeah, no, I'm just doing it because I enjoyed it. I
Yeah.
enjoy it. Then I hope I will be able to to do that in the future. Then of course one gets old, but at least for now, it's exciting and I have fun. So yeah,
Yeah.
I
Also
make
like.
use of it.
Yeah, and I also like sometimes like physics. You can also explain very
Yes,
well with the
definitely,
breakdance,
definitely.
right?
It's really I mean, in a way, I try to use that to explain how proteins interact with each other and they move because finally they do move in the cell, and somehow there are some proteins who are actually which are actually dancing in the cell, and I thought, oh, maybe I can use breakdancing to
Yeah.
explain how these proteins move. But
Yes.
then, of course, talking about proteins is always a bit difficult because people get excited when they connect proteins with bodybuilding.
Yeah.
But
Yeah.
then proteins per se are not really attractive because what is a protein finally? We know, of course, but
Yeah.
then it's a bit difficult to understand the
I
whole
get what
thing.
you
So
mean. So for many people who are doing strength training, muscle building, for them it's just protein one way to build the muscle,
absolutely, yes. So
right?
they get excited when a scientist talks about
Yeah.
proteins because
Yeah.
they think, okay, this is the quickest way to get muscular
Yeah.
mass. But
Yeah.
then they realize I'm talking about alpha helices, and
Yeah.
they're like, what the hell?
Yeah.
No,
Yeah.
and so they come back.
Yeah, no, but it's much more than that. So, in order to actually build your muscle, you need the cell, a muscle cell that actually knows what to do, right? And
Yes, absolutely,
you start
absolutely.
from you need to express the gene that later on helps you actually to build your muscle, right?
Yes,
And
yes,
that is all governed by protein. So in the end, they're all important, and
yeah.
yeah, that excitement is kind of missing sometimes. So I hope that people now appreciate a bit more.
No, the thing is always, it all comes down to the fact we would need to communicate these people complexity. And the fact
Yeah,
this whole thing is more layered than
yeah,
they possibly think. But
yes,
then, of course, then it's easier to just stick to, you know, the protein barrel and the drinks they make
yeah,
to
yeah,
gain muscle or
yeah,
mass.
yeah.
But yeah,
Like Jordi does sometimes.
yeah, like Jordi does.
Yeah.
Ciao, Jordi.
Yes. Yeah, Jordi. Many, many greetings. Yeah, so he does a really good job in explaining that.
Yes, absolutely.
So kudos to him. Let's go to our second part. I guess that would
Yes.
be so people get more excited. Um, So I call the section "Let's break down the protein structures," explaining actually how our cells work. Now so I took out 3 to 4 current reviews that covers multiomics studies. So I'm more on the proteomics side, so mass spectrometry, but also structural biology from your side, Leo. And I would just highlight a bit and talk to you about why is it important to actually go into that direction and why structural biology is so important also for medical applications. So I leave the sources in the comments. But so we both said we just skimmed a bit through, right? So what was like the most prominent information that you took out from the review? So
Well, I think that we can draw, so we can connect how science stepped on in the past with what we are trying to do now. Because every time we went through big revolutions in the past, we blended together different disciplines in science to
yeah,
generate new ones. And I think we are doing this even when it comes to studying the omics.
yeah,
So to understand the foundations of new disciplines, of course, scientists need to focus on the basics, and so they need to understand the proteomics, how proteins work, and the genomics, how genes are regulated. But then, in the end, when they start to build up things and understand how these things work, then we come to a point in which we want to blend everything together and to find a melting pot and to understand what comes next. So I think the point of studying all these disciplines together is to understand how genes interact with proteins, how these things regulate, of course, gene expression, but also the functioning of proteins. And as we said before, things get really complicated when they're put together. So the
yeah,
idea now is to understand how these processes influence each other. And who knows? The point I think is that we want to know more. We want to explore. We want to explore further and to study. And to discover what's the new frontier, what's next,
yeah,
what is beyond all these disciplines,
yeah.
and so that's why we are trying to find connection points between these.
Yeah, and as I said, so structural biology is very, very important to understand the function of a protein and also like sometimes understanding how this protein interacts with its partners.
Yes. Yes, but even
Um,
that structures is already something. I mean, of course, it's important to understand structures, but we are already looking beyond because the structure of a protein. I mean, it's like a statue. Um,
it's
to
a
understand
snapshot.
how, yeah,
Yeah,
it's like looking at a statue to
hmm,
understand how the human body works. The
yeah,
statue is just frozen, while the
yes,
human body, as you said before,
yeah,
is subject to physical laws. If a human body can do breakdancing,
yeah,
it's because it's not a statue.
yeah,
So yes, we are looking at protein structures, but then we need to remember these structures, these statues, they always represent their model. They
yeah,
represent something that is always moved, that is active in a cell. So that's why we're trying to blend many information
yeah,
together using AI
yeah.
to understand how these structures really work, how they function in the
Yeah,
cell.
yeah, that's true. And this multiomics era is also something where it leads or blends into the so-called personalized medicine, which means,
Yeah.
and I find this very well said in the article. The problem that we have is we don't have, as you said before, a one size fits all, right? So every one of us, you, me, and the audience will have a completely different genetic
Yeah,
background.
yeah, yeah.
They will have different lifestyle factors and so on and so on. The exercise plays also a big role. Are you more on the sedentary side? Are you more active, etc., etc.? So to understand why people are more susceptible for one condition of a disease or the other condition of a disease, of course, we have to understand the DNA differences. So that's on the genomic side, right? That we evaluate what kind of risk factors people might have, and that is already part in the clinics. But what I found very interesting, and I'm trying to find this, is the cost. So it was written in the article that genome sequencing costs have plummeted steadily from $1 billion to sequence the initial human genome back in 2000, right? And now, 13 years back, or even more, because in 2022, we only had for one single person $100. So that's already giving us like how crazy that is.
Yes, no, I think this is what happens to any technology in our times. So, like what happened with computers,
Yeah.
we're just finding new ways to put everything up. I mean, to put everything together and make computers and devices that are always more and more efficient
Yeah.
in doing their jobs. I mean, there's this. I don't remember the curve, but there's a curve that describes how fast the microprocessors of a computer more low. I guess you can apply this same law more or less to many many technologies
Yeah.
nowadays. As well as you said, for sequencing and well, what we are seeing with artificial intelligence. I mean, we are literally using it for so many things. And one big innovation that was brought by Google
Yeah.
at the end of my PhD, actually in 2022, was that you can use AI to understand from many different
Yeah.
data, from many different sources,
Yes.
how a structure behaves, and how it can be modeled from the sequencing
Sequence.
data.
Yeah.
So just from the sequence, and from the sequence of amino acids, so
Assets. Yeah.
the building blocks of a protein,
Yeah.
you can understand exactly
Yeah.
how this protein will work and how it's shaped. And this is really exciting, of course. But then there's even more, because
Yeah.
there are so many proteins out there, and we don't know many of them, and we're getting so many data from many different fields of biology that can be just poured
Yeah.
into the artificial intelligence to get more structural data
Yeah.
in return.
Yeah.
And then everything translates to functional data, so you can understand how the system works and how the cell works ultimately.
Yeah.
So you want to use that to, well, eventually the aim would be to, go towards the personalized medicine and find a cure that is really made for you and for treating your particular conditions. And your genome finally.
Yeah. Yeah. And
So I guess this is the direction, and we need to go that way. We need to go on that direction, I guess, and
yeah, I
we
think
need to move forward.
yeah, and if you look like 10, 20 years back,
Yeah.
if you look at the theses of now our professors, sometimes it was just enough to sequence one genome, right? And you got your
Yes,
PhD.
absolutely. Well, like what
Or
they
one
did.
structure you
Yeah,
solved, and
no,
that's
like
it.
what they did decades ago.
Yeah.
So a PhD student was just cloning a gene
Yeah.
in 3 years during their
Yeah.
PhD. Now you can clone a gene in a couple of days. I don't know, even even less. I don't know.
Yeah. Yeah. Usually a week or two, and then
Yes, sure, sure,
usually you
sure.
have it. And I think people who are now doing their PhDs, and for me, instance, doing a postdoc, we are we need to be open to let in other disciplines to understand what we are
Yes,
looking at.
absolutely. That's why we need to communicate
Yeah,
with many different people coming
yeah,
from many different disciplines,
yeah.
and
Yeah, and
to
collaborations are the
yes,
best way. And if you look at the very big papers that find new or novel insights, sometimes they're like an author list of 50 people also,
yes,
it's a lot of work, and we need to foster
coming
this.
from different fields in
Yeah,
science. So
yeah. Yeah, because we cannot be an
chemists.
expert. Yeah, we cannot be an expert in every field, right? So
Yeah.
we need someone from the chemistry side, we need someone from the physics side,
Yeah.
biology. Biology has many different disciplines, so we need people
Yes.
who are
Yes.
specialized there. So it's not that easy. And if I think of what the multiomics paper in general does, it also talks about epigenomics, which is also part what you have done. So, as you pictured really beautifully, so our DNA is of course not. I always say swimming around in the nucleus, being free, and it's tightly packed, condensed, and it can be modified, which is really cool. So we need to modify to actually open up or actually
Yeah, we can
close it a
open
bit
up and
more.
close different areas,
Yeah.
and
So like,
I mean, it's like a book, right?
yeah,
So you can open the book
yeah.
on the pages you're interested
Mm,
into, but then you can close these pages
mm.
and open up other pages according
Yeah.
to how you distribute these
Yes.
bookmarks we were talking
Yes.
about
Yeah.
earlier.
Yeah.
So
And
that's it.
these modifications that happen on the DNA, but also on the so-called histones that actually wrap around the DNA,
Yeah.
are more and more important. And still, we don't understand how they actually, switch certain modifications, or
Hmm.
when does the cell decide to do this and this, and that also has an impact on, does the cell now progress in the cell cycle stage, right? So
Hmm.
um,
Hmm.
is it somehow at this point where it now needs to segregate the chromosomes, so we have mitosis, and all these kind of little things are still mysteries to us. It's not like
Yeah.
that we know everything, and that's why basic sciences yeah, need to be explored further. But I think we also should start using our knowledge that we have from the basic sciences to apply to medical questions as well.
Absolutely. I
Yeah.
think one of the things that are that is really interested in being involved in a multidisciplinary collaboration is that there are these people that are coming from different fields and that are looking at the problem in a novel way from their
Yeah.
point of view.
Yeah.
And this is really interesting because they can offer insights you could never think of. Just because you're used to look at a problem from your point of view, with your own knowledge
Yeah.
that comes from your field, which has been sitting there for ages before
Mm.
you.
Mm.
While on the other side, there are people who are just there for the first time, coming from a different environment, and
Mm.
they're really bringing novelty. So that's why I think it's important.
Mm.
And I guess you can apply the same thing to many different fields.
Mm.
Whenever you have the opportunity to collaborate with a professional who is doing similar things, but from a different point of view.
Mm. Mm. Yeah, totally agree. And to be more on the protein side, so proteomics is kind of covering like what is happening on the protein level. So when we have our condensed DNA and we want to express something, so um, let's say you opened that
Hmm.
specific book that you want,
Yeah.
and you have that page
So, in
open.
order to open the book at that particular page of your interest, what happens is that so you've got these histones
Yeah.
that you can. So basically, these are proteins which behave like I don't know, like some stools, that you used to wrap your DNA around. So in this way, when it's done hundreds, thousands of times, the DNA is really condensed and tightly packed in the nucleus. So what you were mentioning earlier is that when these proteins are modified and they are labeled with different chemicals, they they can open or close this packing of DNA. So when the DNA is open, then the DNA is readable, and of course the instructions that are part of this region are visible and they can be expressed. So the proteins are produced. While when these regions are closed, because they are of course labeled with different chemicals, then it means the DNA is not readable, and it's like a part of the book you just seal, so it cannot be readable, or you cancel out, those forbidden books that are completely blacked out with black ink,
Yes.
something similar. Sometimes our cells are instructed to do that from the beginning, because there are some parts of our genome that are dangerous, so they can be harmful for the cell. And so from the beginning, we know that, and the cell knows that, and prevents itself from getting damaged by using the right chromatin modification, the right labeling, let's say.
Yeah. Yeah.
So at least it can't read that part, but then of course it wants to read other parts,
Yeah.
and so this whole thing is called epigenetics, and the way I usually talk about that is that somehow it's the way we can interact with our genome. Because, of course, it's based on our choices, it's based on how we want to live our life. If we stick to a particular diet, if we exercise, if we do something over the years of our life, we tell our body, we tell our cells to be active on those modifications and to open special chapters of our DNA, and so to actively read some parts of our DNA. I know it looks like, it's kind of like an happy ending and a way to make exciting something that is really, it's all about molecular stuff that is handled at the
Yeah.
level of our DNA. But finally, there are some tweaks we can actually make at the level of our cells that can change the way
Yeah.
we live our
Yeah.
lives.
Yeah.
Um,
We have
it
seen
all
that.
comes.
So, like
Yes,
for
yes,
diabetes, for
absolutely.
instance, people who have type 2 diabetes, if they change their lifestyle, if they change their diet to a way where they can reduce again their blood glucose levels and get maybe their insulin resistance a bit down so that they are more responsive to that. That has been shown in some of the
Yes,
studies already that if you change through the diet and through the exercise level, that this has an impact. And that goes back to the epigenetics, right? So
yes,
the
absolutely,
marks
absolutely.
have been changed in such a way that you respond better and that you can mitigate your disease a bit.
Yeah, let's say the moral of the story would be that we cannot change our genetics, because of
No.
course we inherit that from our parents,
Yeah.
and we need to live with that, but we can change the way we read our
Yeah.
genetics. So we can change, we can decide to avoid some chapters of our DNA and read others instead. It all comes down to the way we want to live our life, and this is, this is a nice happy ending, I guess.
Yeah.
It's not that it's not easily achievable, because of course it has to do with the way you live your life. It's a whole bunch of choices you need to change. But then, of course, I mean, if you quit smoking, for example,
Yeah.
there are some parts of, well, of course, your lungs will thank you in the end, but then there are some parts of your genome that are not read anymore. For
Yeah.
example, the ones that are involved in the inflammatory response.
Yeah.
Just because you quit smoking and you don't go through a continuous inflammatory response at the level of your lung. This inflammatory response that is not switched on anymore somehow will benefit your body. And we all know that it's not easy for smokers to just quit smoking. But then,
Yeah.
of course, it's up to you. You can change the way you read your DNA. You just need to go
Yeah.
through that.
True. And also when we take the diet aspect, so of course what we eat matters. So
Yes.
if it's
Yes,
whole
absolutely.
foods, fruits, vegetable, etc., etc., I don't need to go on this. Margarita had that in her episode already, but also the what kind of nutritions we have available for the cell is also important, which is covered by metabolomics, for instance, that we
Yes.
measure
Yes.
and analyze. And what I find very interesting, and now many people go on this topic, is like the gut microbiome and gut brain axis, gut muscle axis, whatever axis. And there we need to be a bit careful because, of course, there are studies out there saying that, for instance, people who drink diet soda because they use the artificial sweeteners instead of sugar, for instance, that the gut microbiome changes. But for that, we don't know if it's in a good or
Oh,
negative way.
that's also interesting. I mean, it really doesn't have it's not directly connected with the proteomics, but also the
Yeah.
way that what influences our
Yes.
the bacteria in our guts is really interesting because these bacteria really interact with many processes
Yeah.
that are part of our body, and so
And they
um,
produce the
yeah.
metabolites sometimes,
Yeah,
right? That
yeah,
are
absolutely,
then taken
absolutely.
up by
Yeah.
our cells that have a respond on a protein then,
Yeah.
like that connects it. But it's an indirect response that we
Yes,
have. And
absolutely,
we don't
absolutely.
really understand how this interaction or how this, yeah, how this works actually. And we need to try to figure this out. And this is also something that will be part of the future studies, having that also combined. And in the study, there was something mentioned like wearable devices. So, of course, we as basic researchers are not really in touch with wearable devices. That would be more onto the medical professionals who have these blood pressure devices that all the time take your blood pressure, or how your sleep is, or all these
Yeah.
kind of things, or even what is your blood sugar level right now, right? So we are not into that, but I find this very interesting that they said that this is also part of the multiomics, because when I think of it, multiomics is for me like everything that I can measure in the cell and doesn't have to be a device externally somewhere. So I find this
Yeah,
very
but
interesting.
finally, data can also come from many different devices. And if you're going to pour all these data in an artificial intelligence,
Yeah.
ultimately, to combine them all and find out some correlations, you will probably need to collect data, throughout the day and getting different profiles according to particular situations in
Yeah.
which you can be exposed.
Mm.
It can definitely be interesting.
Let's have a thought experiment here. Let's say we have taken all the data of the whole entire world population variables, like multiomic studies, and feed it into an AI. Would you say we need scientists later on to interpret it, or will be the AI taking?
Well, the idea is that you always need scientists after the data have been processed, because it's like when you're trying to figure out what you want to say in a post or at the beginning of a podcast, for
Yeah.
example. You try and ask the AI, and the AI comes back to you with an answer, but then you want a human being to decide what's the best pitch, or what's the best idea, what's the best example that you can use for what you know is your final product or your research. Because AIs can speed up the process, and they can propose a variety of solutions, but finally the idea is that they're a tool, and so they need to be used by a scientist. They cannot replace scientists. They cannot replace the human mind that is there just
Yeah.
to choose carefully. So the idea would be to use them. The idea would be to use them to enhance the possibilities we have on the plate, and to get more answers. But then
Yeah.
finally, the final answer should be chosen by a scientist, should be chosen by us.
Yeah, because when Martin was here as a guest, he read an article about one of the people from Google DeepMind,
Yeah.
and he was telling that the hypothesis-driven research will not
Yeah.
exist anymore. So
Yeah, well, I don't know, because the thing is that, well, if there are people who are asking themselves these questions, I think they are important, and they need to be discussed. But at the same time, from my naive point of view, because of course I'm not an AI expert, but I think there are many hypotheses, right? So you finally need to choose one.
Yeah.
The AI can come to you proposing a variety of hypotheses, a variety of paths you can dive into after you find out something. But finally, the ultimate decision relies on you, right?
I would say so true. Yeah. So I would like to judge. I don't want to have like already the answer in front of me. I would like to have the freedom to pursue what I want to do and what to
Yes.
want to explore, and not an AI tells me, no, the great hypothesis is now this.
Because even if the AI is really informed, and it knows what's the most feasible path, so it will come back to you, and it will tell you, I know that we've got these 100 different choices, but I can tell you this one is the best one, because
Yeah.
you don't have enough money at the moment to go through this way. So what are we going to do?
Yeah.
I can always choose, right?
Yes.
And then of course it will be up to me to show I can make it.
Yeah.
But it's finally up to me. It's it's the human being who decides, right?
Yeah.
Or at least this is
Completely agree.
this
And
is what I believe.
yes, and if you have this integrated multi-omics studies, and if you don't have medical professionals inside these, let's say, decision makings, that's only one part, right? You also need the other part because the clinician aspect, is not there yet in the multi-omics study. So
Yeah.
you would need a medical professional to and the experience that someone has, you cannot train an AI on that, right? So
Yeah,
someone
absolutely.
comes in. So I think AI, as you said, is a great tool, and it is already used a lot, and we see it in many, many research
Fields.
fields, and also in structural biology, as you said. So Google DeepMind's AlphaFold was like a huge thing, which is still a huge thing, and I there was an article where people said we don't need structural biologists anymore, which I don't believe. We still need people
Well,
who
they used to say
understand.
that they used to say we don't need crystallographers
Yeah,
anymore when the cryo-EM came
yeah,
out.
true.
Crystallographers are still
Are
there
still there.
simply because it turned out cryo-EM is useful when you've got big complexes, big proteins,
Yeah.
but then the detail that crystallography or NMR can
Mm.
provide is unmatched.
Yeah.
So
Yeah.
then all these data can be taken all together and fed to an AI so
Yeah.
it can put everything together and find
Mm.
something new. So
Yeah.
finally, you're still using a lot of stuff from the past years.
Yes.
And you know, all these titles can be exciting. You know,
Mm-hmm.
you don't need scientists anymore.
Yeah.
But finally, isn't that connected to what we were talking about earlier?
Mm.
The fact that there are these big titles
Mm-hmm.
with these big promises, but
Yeah.
they just ignore the
Yes.
complexity behind.
Yes.
So
Yeah. Yeah.
I
No,
wouldn't mind them.
completely. Yeah. No, no, no. But I completely agree, and it's a shame that that we sometimes are narrowing it down so much, right? That it gets so watered down, and we cannot appreciate the complexity sometimes
Yep,
of biology with
absolutely.
that. And um, in my years of doing research, I had the opportunity to do a bit of crystallography and a bit of cryo-EM, and I totally understand to get to that structure is also a bit of luck, but also a
Yes,
bit of
absolutely, absolutely.
yeah, you have to try around a lot. And sometimes it does not work for whatever reason. And these are all stories that we kind of, yeah, tend to forget,
Yeah.
how much work it is to generate the data and analyzing the data. And
Yeah.
currently in my project, when I do proteomics. So I have a variety that I can apply. So I want to quantify the proteins that are different in each condition, but I also have the bridging the gap between structural biology and proteomics by using cross-linking mass spec. And sometimes that people helps a lot because they suddenly understand how the subcomplexes of a protein are interacting with each other, which they did not know before because they were just purifying the complex but not getting any structural insights. And then suddenly they know, okay, this one is very flexible or labile or just gets off, and it's really interesting how the structure of complexes determine the function of the protein, and also interchanging the
Yeah.
subunits to make and form new complexes. That is very fascinating, and maybe we just talk about one of the other which I review, which I really liked, is Understanding the Cell, Future Views on Structural Biology. So there they were talking about all the different methods that we know for now. You have not been in the field so far anymore, but still you're with your colleagues in contact. Um,
Yeah.
can you maybe give some insights what is kind of new besides, of course, AlphaFold, but also where is structural biology heading?
Well, I guess the future is to just put all these as I was saying before, is just to just put all these actors, these proteins in a context. So the idea is to study all these proteins in their cellular context, picking up data from many different fields and feeding them to AI so they can make sense and they can help structure they can help building up a protein structure. That would be the future. And at the same time, there are many techniques based on cryo-EM that can allow you to look, well, they are sometimes already in use, and they can sometimes look at the proteins in their natural environment. So it means looking at proteins directly from the cell. And
Okay.
that makes a perfect sense, because we were talking about the fact that proteins are just statues of something that is moving, that
Yeah.
is active, and the way you want to look at them is how they actively, how they actively function. They work in a cell. So to get pictures of them while they are in action, so you can understand better how they work. I think this is the future. It would be to get many different data from many different fields, and to get them all together, to make sense of new structures, to understand also how structures really work in the cell.
Yeah, completely agree. So, I yeah, sometimes it's a bit difficult to understand why a certain protein is behaving as it is, right? And if you don't know how it looks like, it will be difficult to grasp. But since structural
I mean,
biology, yeah,
it would be like studying an animal in a zoo,
yeah,
and um, trying to
it's not
understand
in a
how.
natural environment,
No,
right?
of course, and trying to understand how it behaves, from an environment that is kind of controlled,
Yeah,
and there's. I mean, it can help, but it's not the real thing. So
yes,
that's why you want to move to cells to understand how the things
yeah,
work.
yeah,
And of course, the reason why we didn't do that before is that because the technology
was
wouldn't
not there.
allow it in the
Yeah.
past. And now we've got all these tools that we can use,
And
and
all the
bring together.
researchers
Yeah, yeah, yeah,
who
yeah,
were frontiers who actually made it happen that we have these technologies,
yeah,
right?
absolutely.
So without them, so in my field, in the proteomics fields, it's Rüdi Ebersod, for instance, or Matthias Mann,
Yeah.
are the forefront people in proteomics, and many others as well. But of course, without them, the field wouldn't be there where it is. And same as for structural biology, for the people who did improve the cryo-EM, for instance. You said it was already there in the 70s, but it was not good enough.
No,
And now,
it took us I don't know, more than 40 years to get
yeah,
the results we've got today, and we're still
yeah,
improving. So
yes, yeah,
that's it.
completely. And um, I'm really excited to be a part of this, of this era, let's say, and be actively doing my research, of course. But
And talking about that in a podcast,
yeah, yeah, and
you're contributing to letting people know about the progress
yeah, and
of
it's
science.
sometimes such a shame that so there are so many great articles out, but not covered by people because it's, like no one
Yeah, I guess
cares
this is the
except
struggle
us.
of anyone in
Um,
research,
yeah,
or yeah, no, this is also, I mean, it's another problem. The fact we are literally filled with information coming
mm,
from many different sources, so
mm,
it's always difficult to keep track of everything. And I mean, it's hard for us that we
mm,
know the field, and of course, it would be even harder for people who don't really know what's the best thing
mm,
they need to focus on. So
yeah,
of course that's the reason why science communicators are there, to help them,
yeah,
or at
yeah.
least to
At
try to help
least
them.
they should, try to help them. Um, and yeah, I think we covered a lot from most of the parts from the reviews, I guess. Or do you have something to add?
Uh, no, I think I've never been in a podcast for so long, so
I'm sorry.
I'm already astonished. I'm
Okay.
still alive and I'm
So
not
come on.
fainting.
Uh,
So I don't know, if you feel we can go on and discuss something more. I'm completely available, and we can go on. But then No,
I think the most
I,
of it we covered,
yeah,
right? So
yeah, yeah,
what the
absolutely,
field is
absolutely.
about, and of course we don't want to spread misinformation at this point.
Not at all, not at all, not at all.
So I think it's also a good closing mark, and I at this point I always ask my guests, since we're coming to the end of the episode,
Yeah,
if you would like to say something to the audience, so that the
to the
stage is yours.
audience. Well, if you if you are listening to this podcast just because you're interested in science, well, let's say, I'll rephrase that. If you're listening to this podcast because you need to find out who you want to become in life, and you feel inspired by science and you want to find out more, then never stop asking yourself questions. I mean, even if you already found out what you want to be in your life, I think the best idea would be to never stop asking yourself questions, and always getting involved in new adventures, new projects, and of course, it's what I'm telling to my students too. Never stop learning, never stop being enthusiast. And this is really what makes the difference, and what allows us to do more.
Yeah.
And I think AIs will never be able to do that, even if they are built to interact with us and to help us. They will never be able to be as curious as human beings. So we are born to be curious. Just stick to it, and
Yeah.
be passionate about what you're doing. I
Yeah.
think this is the best take-home message you could pick from this conversation I had with Chandni. And I think it's always nice to remember this, whatever you're doing in your life.
Thank you so much for your wise words, your wisdom you're sharing.
Oh, come on,
Um,
it's
maybe just, of course, being open-minded is something that I would
yes,
also,
absolutely,
yeah,
absolutely,
you know.
absolutely.
Okay, so we are at the end of our conversation with Leo. First of all, thanks a lot, Leo. We had in the beginning some technical issues, but I'm
No,
really
I'm so
happy.
sorry about that. It was the first time I was using this setting in ages. So
Yeah,
thank you for being patient with me, and thank you so much for inviting me to
yeah.
this
No,
podcast. I
really,
really had fun.
I really appreciate your work on social media and also on YouTube, guys. So please follow him on Instagram at Leo Feletto, or Leo is on YouTube. So I will post the link down. So we communicated about many things. So we had why he is so enthusiastic about science, how he got into science, and now he is giving this excitement as a teacher to his students, but also is a very passionate science communicator. And now we had a little journal club as well, so that you learned how important structural biology, but also multiomics studies are, to actually understand a bit more and how we can translate this knowledge later on for medical applications. I think you know that I have a little slogan at the end of my
Yes.
thing of my
Would you be so
podcast, or
kind to
in general.
remind me about that?
Yes,
Because I think I forgot it.
yes. So let a little dose of honest science in your life and brighten up your day. So the question
Okay.
is, if you would be up to tell that slogan with me.
Absolutely, yes.
Okay,
I just
cool.
need to write it down so
Yeah,
I make sure I say it right.
yeah.
Let me see. Notes. All right. Now it's there. Oh, come on. Oh my days. What is happening? Oh, we
I don't
can.
know. I sent you the video script, so it's at the end of
Yes,
the
no,
screen.
it's just
Very
okay.
slow.
Let
A
a
little
little
dose,
dose of
honest,
honest science
yeah, in your life
in your life.
and brighten up your day.
Yeah,
Okay, cool.
brighten up your day. All right,
How
excellent.
do you like the slogan?
It's okay. It's positive, so it gives positive vibes, and
Okay.
yeah, maybe we can shorten it. I don't know. No, but
It's already
it's okay. It's
branded.
okay. It's okay. It will work. It will work fine. Don't worry about that.
Okay.
Let a little dose of honest science in your life and brighten up your day.
Yeah. Genau.
All right.
Perfect. Okay. So 1, 2, 3. Let a little
Let a
dose
little dose of
of
honest
honest
science in your
science in your
life
life and
and
brighten up your
brighten
day.
up your day.
Awesome. Cool.
Okay.
Thank you
Cool.
so much, Leo.
Thank you. And
Yeah. So guys,
anytime, really, if
yeah.
you want to discuss something more in the future, I'm really open. And at the same time, next time I will be ready, so my setting will work from
So
the
yeah,
beginning.
people need to subscribe, right? And need to leave a like and maybe a little
Yes,
comment.
absolutely, absolutely,
I
absolutely.
would love to see everyone in the comments maybe spamming that we should have a part 2 with you about some fascinating science.
Hopefully.
Um, Maybe last question. Do you have a
Yes.
favorite structure?
A favorite structure.
Yeah.
Oh, so at some point in my PhD, I was working with this chromatin modifier that is called P300, which is a
Hmm.
really complex protein. I mean,
Hmm.
if you look at the structure, that is part of it is predicted by oh Jesus, by AlphaFold,
Hmm.
that is predicted by AlphaFold. This is an intrinsically disordered protein, so it's like a spaghetti monster.
Hmm.
Oh yeah, it makes sense. I'm Italian, so spaghetti protein. Yeah. No, but you can see this protein. I mean, I struggled so much to purify it, and in this predicted structure, this protein is just all over. You can see these spaghetti filaments going all around of it, and it's so beautiful. So every time I think of B300, I'm like, wow, that's a protein.
And when
That's
you
a
eat a spaghetti bowl.
yes, of course. I think of B300 all the time, all the time.
Okay, guys, have a wonderful evening. Hope you enjoyed our conversation, and we see us in the next video.
See you
Bye.
next time.
Daily dose of honest science.